Good morning, students. Today we are going to see the factors affecting bacterial growth. In the last classes we have seen, that is in the previous class we have completed the growth. What is growth? Growth is nothing but increasing in the size of the organism as well as a division of the organism into many cells. This is called growth. So in the last class we have seen the growth, and growth curve also we have seen, that is lag phase, log phase, stationary phase and decline phase. After that we have seen the types of fermentations, that is your batch culture, continuous culture. In that continuous culture we have seen two things, that is chemostat and turbidostat. Next, after that we have seen your next one is your synchronous cultures. After that we have seen how to measure the microbial number as well as microbial mass, that is cell density. That is how to measurement of cell number and cell mass of microorganisms we have seen. Is it okay? The measurement of microbial growth, that is by means of cell number and cell mass we have completed. Now today we are going to see the factors affecting your bacterial growth. What are the factors affecting bacterial growth? Are generally many factors in the environment, they will control the growth of the microorganism. Of them, the first one is temperature. All of you know temperature. Temperature plays an most important role in the growth of the microorganisms. In the growth of the microorganisms, temperature plays a major role. Then that means temperature affects the growth of the microorganisms. Generally, at very low temperature, microorganisms do not grow. At very high temperature also microorganisms do not grow. What happens at very low temperature and at very high temperature? See here, at very high temperatures, microorganisms, enzymes, proteins, see here, proteins, enzymes, next their cell wall components, all these they get damaged. See here, at high temperatures, the denaturation or disintegration or damage of its proteins, enzymes as well as cell wall components, all these will take place, so that the bacterial cell will die. We are talking about growth, but this will lead to high temperature, will lead to the death of the microorganism because of the degeneration or disintegration or damage of your proteins, enzymes, hormones, lipids, cell wall components, etc. All these get damaged, so that the cell will ultimately die. This happens during high temperature. Then at low temperature, what happens? At low temperature, all these are there without any disturbance. They did not get damaged, but their function will be stopped. Whatever it may be, at low temperature they do not show that type of function which they have to show. At high temperature, they cannot show their function which they have to show because of their damaging. That means at high temperature, all these things they get damaged, deteriorated or degenerated or denatured, and at low temperature they do not show their properties properly, they do not show their function. For that reason, at low temperature and at high temperature, bacterial growth is stopped. Now here, then what should be the temperature? The temperature for the growth of microorganisms, that is bacteria, is 30 to 40 degrees centigrade. Generally 37 degrees centigrade is the optimum temperature for your bacterial cells, and for fungal cells the temperature is up to 20 degrees centigrade. That means at 15 degrees centigrade to 20 degrees centigrade, the fungal organisms will grow properly. That means 20 to 40 is the temperature at which the microorganisms generally grow. Now coming to the microorganisms, based on the temperature, the microorganisms are of three types. They are A. Mesophils, B. Thermophils, C. Psychrophils. Now coming to the mesophils. These are the organisms, microorganisms. These are the organisms that grow at a temperature of 20 to 40 degrees centigrade. They grow at a temperature of 20 to 40 or 45 degrees centigrade. Now coming to the thermophils. The name itself indicates that the mesophils, they stay in medium temperature. That is in room temperature. Now thermophils, these are the organisms that grow at very high temperatures. Thermo means temperature. Thermos flask means the flask in which you will be pouring your milk or coffee or tea, and that will be stayed for long periods, they will be hot only. So thermophils means because of that only it is called as thermos flask. Now these thermophils, they can grow at 80 to 100 degrees centigrade. These organisms, they can grow at 80 to 100 degrees centigrade. They can withstand 80 to 100 degrees centigrade in other words. But these things, you can generally see them in hot water springs. In hot springs, or you can call them as geysers, these are grown. In hot water springs, they grow. Now psychrophils. These are the organisms which grow at below 4 degrees centigrade. That means -0 to below. That means at very cold temperatures, cool temperatures, they will grow. They are called as psychrophils. In Arctic and Antarctic areas, they will grow. In Himalayan areas also they will grow. These are the different types of organisms based on the temperature. Coming to the second one, pH. What is the definition of pH? pH is a negative logarithm of hydrogen ion concentration. Now this pH is there. Generally pH also plays an important role in the growth of the microorganisms. Then what is pH? Generally the pH should be alkaline, acidic. All of you know that pH 7 is neutral, pH above 7 is alkaline, below 7 is acidic. All of you know that 7 is the neutral point, that is your drinking water will be 7, pH is 7, and if it is consisting of large amount of bases, it is known as alkaline or basic in nature, and if it is below 7, the pH is known as acidic in nature. Okay. Now see here, this pH, based on the pH, the organisms, they are alkaline organisms or acidic organisms, basic or acidic organisms. Now coming to the pH of the organisms, that is alkaline pH is for bacteria, and acidic pH is for algae and fungi. But for both algae and fungi, the nature of the bacterium is acidic. The pH of the bacterium is acidic for both algae and fungi, and alkaline for your bacteria. Okay. Now see here, these organisms, they will grow at this pH. Now coming to the natural things, generally what happens is, when you are having sulfur bacteria, its pH is 0. That means highly acidic in nature. దీనికన్నా acidic ఇంకా లేదు. This is 0. its pH is 0. That means highly acidic. Now అంటే 0 to 7 వరకు చూసుకుంటే acidic అని, 0నే దాని pH. Now urine infection causing bacteria, its pH is 11. That means more than 7, it is highly alkaline. Now your pickles in food industry, the pickles are stored and at a pH of 3 to 4. That means it is highly acidic. అంటే ఏంటి, మనకి ప్రియా పికిల్స్ అట్లాంటివి ప్యాక్ చేసినప్పుడు దాని pH ఎంత ఉంటుందంటే 3 to 4, highly acidic in nature. నెక్స్ట్ సల్ఫర్ బ్యాక్టీరియా, ఇట్స్ pH is 0. నా యూరిన్ ఇన్ఫెక్షన్, ఎవరికైనా మనకి యూరిన్ ఇన్ఫెక్షన్ ఉంటే కనుక వాళ్ళకి pH 11 ఉంటుంది. అందుకోసమని వాళ్ళకి యూరిన్ ఇన్ఫెక్షన్ ఉన్న వాళ్ళకి సిట్రాల్కా అని ఒక సొల్యూషన్ ఇస్తారు డాక్టర్స్ తాగడానికి. ఆ సిట్రాల్కా ఇస్తే ఏమవుతుందంటే, లేకపోతే నోవోమాక్స్ వాటన్నిట్లలో ఎసిడిక్ నేచర్ ఉండేటట్టు ఇస్తారు. సిట్రాల్కా అంటే ఆ ఆల్కలైన్ నేచర్ని తగ్గించడానికి సిట్రిక్ యాసిడ్ ఇస్తారు. పుల్లగా ఉండి ఆ సొల్యూషన్ తాగుతారు. మీకు ఎవరైనా పెద్దవాళ్ళకి కనుక యూరిన్ ఇన్ఫెక్షన్ ఉంటే వాళ్ళు సిట్రాల్కా తాగుతారు. ఎందుకంటే యూరిన్ ఇన్ఫెక్షను ఆల్కలైన్ బ్యాక్టీరియా వల్ల వస్తుంది కాబట్టి దాన్ని ఎసిడిక్ సొల్యూషన్ సిట్రాల్కా తాగడం వల్ల సిరప్ ఆ ఎసిడిక్ నేచర్ వల్ల ఈ ఆల్కలైన్ నేచర్ న్యూట్రల్కి వస్తుంది, 7కి వస్తుంది అని. సో దిస్ ఈజ్ యువర్ డిపెండ్ ఆఫ్ ది pH. కమింగ్ టు ది థర్డ్ వన్, ఆక్సిజన్. See here, coming to the next one is your oxygen availability, or you can call it as oxygen availability. We'll talk it as oxygen next one is oxygen availability. The next factor for the growth of that controls, that affects the growth of microorganisms is oxygen availability. All of you know that oxygen is essential for all the living organisms, but there are certain organisms which do not require this oxygen. Such type of organisms which do not, they grow in the absence of oxygen, they are called as anaerobic. All of you must be knowing that since your childhood you must have studied it. But based on the availability of oxygen, the organisms are classified into strict or obligate aerobes, strict or obligate anaerobes. Next, facultative anaerobes. These are the 3 different types of organisms, microorganisms, based on the available oxygen availability. Now coming to the first one, that is strict obligate aerobes. These are the organisms which grow only in the presence of oxygen. In the absence of oxygen they cannot grow. They are very strict, they are sticking to the oxygen availability. If oxygen availability is not there, they cannot grow. So they are called as strict or obligate aerobes. Wherever you get an obligate word in it is a scientific word. Obligate, it means very strict, and in the along with that, if you are adding aerobes, means they can grow only in the presence of oxygen. Coming to the strict or obligate anaerobes, these are the organisms which can grow only in the absence of oxygen. If a little bit of small amount of during their growth period, if at only a fraction of second, for a fraction of second, if oxygen availability is there, immediately they retard their growth or they stop their growth. అంటే ఏంటి అంటే అవి గ్రో అవుతూ ఉన్నాయి ఎనరోబిక్ కండిషన్లో, ఆక్సిజన్ లేదు, ఇన్ ది అబ్సెన్స్ ఆఫ్ ఆక్సిజన్ దే ఆర్ గ్రోయింగ్. కానీ దాని గ్రోత్ మధ్యలో మనం ఎప్పుడో సడన్గా కొంచెం ఆక్సిజన్ ఇచ్చాము తెలియకుండా, ఎరోబిక్ కల్చర్ అనుకొని, ఆ ఫ్రాక్షన్ ఆఫ్ ఎ సెకండ్ చాలు, ఆక్సిజన్ కనపడగానే అది గ్రోత్ ఆపేస్తుంది, ఇంకా మళ్ళీ గ్రో కాదు. ఎనరోబిక్ కండిషన్ ఇచ్చినా కూడా గ్రో కాదు, ఎందుకంటే దాన్ని గ్రోత్ని మనం స్టాప్ చేసేసాము, అంత స్ట్రిక్ట్ అది. నెక్స్ట్, కమింగ్ టు ది ఫ్యాకల్టేటివ్ ఎనరోబ్స్. దీస్ ఆర్ ది ఎనరోబ్స్ విచ్ కెన్ గ్రో both in the presence of oxygen and in the absence of oxygen. See here, they can grow in the presence of oxygen as well as absence of oxygen, that is no oxygen. Both in these conditions they can grow. If oxygen is there, no matter, they will grow. They are anaerobes only, but if oxygen is available, they will grow. If oxygen is not there also, as they are anaerobes, they can grow. That means they can grow both in the presence of oxygen. If oxygen is available, they can grow. If oxygen is absent, they can grow. If oxygen is available, they can grow. If oxygen is not available, then also they can grow. These are the different types of organisms based on the availability of oxygen, based on the oxygen availability. Now coming to the fourth one, that is osmotic pressure. See here, coming to the osmotic pressure. What is osmotic pressure? What is osmosis? The movement of solution from low concentration to high concentration is called osmosis. All of you must be knowing. That is, from low to high concentration, it is called the movement of solvent or solution from low concentration to high concentration is called osmosis. For that, see here, O means sounds like low. So, it is low concentration to high concentration. This is known as osmosis. ఎట్లా గుర్తుపెట్టుకోవాలి? లో అనగానే ఓ లాగా కనిపిస్తుంది. ఓ ఈజ్ లో, హై లాగా లేదు. లో అంటే ఓ ఈజ్ సౌండింగ్ జస్ట్ లైక్ యాజ్ యూ కెన్ ప్రొనౌన్స్ ఓ మీన్స్ ఇట్ ఈజ్ రిథమిక్, ఇట్ ఈజ్ రైమింగ్ టు లో. ఓ ఈజ్ రైమింగ్ టు విత్ లో. సో ఫ్రమ్ లో కాన్సన్ట్రేషన్ టు హై కాన్సన్ట్రేషన్. ఇది కాకుండా మనకి కాన్సన్ట్రేషన్ గ్రేడెంట్ అని ఉంది, వేరేవి ఎన్నో ఉన్నాయి. దానిలో కాన్సన్ట్రేషన్ గ్రేడెంట్ అంటే ఫ్రమ్ హై కాన్సన్ట్రేషన్ గ్రేడెంట్ టు లో కాన్సన్ట్రేషన్ గ్రేడెంట్ అంటారు. అది కాదు, జనరల్ డెఫినిషన్ ఆఫ్ ఆస్మాసిస్ ఈజ్ ఫ్రమ్ లో కాన్సన్ట్రేషన్ టు హై కాన్సన్ట్రేషన్. Now, based on the osmotic pressure, the microorganisms are classified into two types. They are A and B. They are halophils. First one. Second one is your osmophils. See here, halophils means these are the organisms, these are the microorganisms, or these are the organisms which can grow in high salt condition. That means here high salt condition is maintained. That means high sodium chloride, calcium carbonate, such type of high alkaline condition is maintained, where it consists of a large amount of salts. That is, your seawater is seawater and ocean water consists of halophilic microorganisms, because there it consists of large amount of salt, sodium chloride. Because of the availability of large amount of salt content, all the microorganisms, they cannot grow. Only halophilic microorganisms can grow. Coming to the osmophilic, these are the organisms which can grow only in the presence of high sugar content. See here, they can grow only in the presence of high sugar content. Okay, halophils are the organisms which grow at high salt concentration, and osmophils are the organisms that grow at very high sugar concentration. Now, where can you find these osmophils? These osmophils are seen in the sweets. See here, in the sweets, if you keep the sweets, after preparing sweets, if you keep them, if you start eating them for longer periods, అంటే చూడండి అమ్మ, మన ఇంట్లో స్వీట్లు తయారు చేశారు, అది ఎన్నో రోజులు దాచుకొని మనం తింటాము. అట్లా తిన్నప్పుడు ఏమవుతుంది అంటే, దెర్ ఈజ్ అ పాసిబిలిటీ దట్ దెర్ ఈజ్ అ ఛాన్స్ ఆఫ్, బికాస్ ఆఫ్ ది హై షుగర్ కంటెంట్, ఆస్మోఫిలిక్ బ్యాక్టీరియా మే గ్రో ఆన్ దట్ మెటీరియల్. అంటే ఏంటి, మనం తయారు చేసుకున్న స్వీట్స్లో షుగర్ ఎక్కువగా ఉండడం వల్ల దానిలో ఆస్మోఫిలిక్ బ్యాక్టీరియా దాని మీద ఇన్ఫెక్ట్ చేసి దాని మీద ఉండడం వల్ల అది మనం తింటే మన పొట్టలోకి ఇట్ విల్ ఎంటర్ ఇంటూ అవర్ స్టమక్ అండ్ ఇంటెస్టైన్స్. సో దే కెన్ గ్రో ఆన్ దీస్ హై షుగర్ కాన్సన్ట్రేషన్స్. ఇస్ ఇట్ ఓకే? నౌ కమింగ్ టు ది ఫోర్త్ వన్ ఈజ్ కంప్లీటెడ్. కమింగ్ టు ది ఫిఫ్త్ వన్, radiation. See here, this radiation plays an important role in destroying the microorganisms. They will not allow the growth of the microorganisms, rather they destroy the growth of the microorganisms. That means they show a damaging effect on the growth of the microorganisms. What they will do, see here, these radiations, they are UV radiations, IR radiations, gamma radiations, see here, X-rays. These are the radiations which are generally observed. See here, gamma radiations, these are the radiations that are generally used for destroying the microorganisms or for damaging the nucleic acids of microorganisms. Now, these IR, these three, that is your IR radiation, this is A, B, C, D. Okay. Now see here, this IR and gamma rays and X-rays, IR, that is infrared radiations, gamma rays and X-rays, they are useful for destroying the microorganisms. How they will destroy? Especially the, I will write here gamma rays, here I will write gamma rays, and here I will write infrared rays. These two are the radiations which have high penetration power. They have high penetration power and low ionizing capacity. Infrared and X-rays. ఎందుకు హై పెనిట్రేషన్ మేడం మీకు ఎట్లా గుర్తుంటాయి ఇవన్నీ అని అడగచ్చు. ఎక్స్రేస్ ఎక్కడికి వెళ్తున్నాయమ్మా? ఎక్స్రేస్ విల్ ఎంటర్, దే ఫాల్ ఆన్ అవర్ బాడీ, స్కిన్, దే విల్ మూవ్ త్రూ ది ఫ్లెష్ అండ్ దే విల్ రీచ్ ది బోన్స్. ఎప్పుడు మనం వెళ్తాం ఎక్స్రే కోసం? ఏదైనా దెబ్బ తగిలి కనుక బ్రేకేజ్ ఉందా, ఏమైనా ఫ్రాక్చర్ ఉందా అని తెలుసుకోవాలంటే మనం ఎక్స్రేకి వెళ్తాం. ఎక్స్రేకి వెళ్ళినప్పుడు ఏం చేస్తాడు డాక్టర్? ఎక్స్రే మెషిన్ దగ్గర ఇట్లా పెట్టగానే దీని మీద ఎక్స్రేస్ పడి స్కిన్ నుంచి ఫ్లెష్లోకి వెళ్ళి ఆ ఫ్లెష్ నుంచి బోన్స్ దాకా వెళ్తున్నాయి, అంటే ఇటు లోపలికి పెనిట్రేట్ అవుతున్నాయి. ఎక్స్రేస్ హ్యావ్ హై పెనిట్రేషన్ కెపాసిటీ, అందుకే వాటికి లో అయనైజింగ్ కెపాసిటీ, లో వేవ్ లెంత్. హై పెనిట్రేషన్ ఉంటే లో వేవ్ లెంత్ ఉంటుంది, అందుకే లో వేవ్ లెంత్ ఉండడం వల్ల, బరువు తక్కువ ఉండడం వల్ల లోపలికి దూసుకుని వెళ్ళిపోతుంది. అదే కనుక యూవీ రేస్, మనం యూవీ రేస్ ఎందుకు అనుకుంటాము? సన్ స్క్రీన్ లోషన్స్ అవన్నీ ఎందుకు రాసుకుంటున్నాం మనం రోజు ఎండలోకి వెళ్తే కనుక ఆ సన్ను పడి దానిలో ఉన్న యూవీ రేస్ మన మీద పడితే మనం డార్క్ కాంప్లెక్షన్లోకి అయిపోతాము, లేదా మన స్కిన్ ఎలర్జీస్ వస్తాయి, స్కిన్ ర్యాష్ వస్తుంది అని చెప్పి మనము సన్ స్క్రీన్ లోషన్స్ రాసుకుంటున్నాం. ఆ సన్ స్క్రీన్ లోషన్స్లో అన్నీ ఏముంటాయి అంటే యూవీ రేస్ని రిఫ్లెక్ట్ చేసేవి ఉంటాయి దానిలో. అంటే ఏంటి? యూవీ రేస్ సన్ నుంచి ఇలా పడగానే అవి రిఫ్లెక్ట్ అయ్యి అలా వెళ్ళిపోవాలంతే కానీ మన మీద పడకూడదు. అందుకోసమని సన్ స్క్రీన్ లోషన్స్లో అవన్నీ ఉంటాయి, అందుకోసమని మనం రాసుకుంటాం. అట్లాంటిది యూవీ రేస్ దే ఫాల్ ఆన్ ది స్కిన్ అండ్ డ్యామేజ్ ది స్కిన్, స్కిన్ ర్యాష్ వస్తుంది, ట్యానింగ్ అవుతుంది అనుకుంటాం, స్కిన్ ట్యానింగ్. అందుకోసమని యూవీ రేస్ హ్యావ్ లో పెనిట్రేషన్ కెపాసిటీ, అంటే లోపలికి వెళ్ళట్లే పైనే ఉన్నాయి, అందుకే హై వేవ్ లెంత్. ఓకే, దే హ్యావ్ హై వేవ్ లెంత్, యూవీ రేస్ హ్యావ్ హై వేవ్ లెంత్ అండ్ లో పెనిట్రేషన్ కెపాసిటీ. ఎక్స్రేస్ హ్యావ్ లో వేవ్ లెంత్ అండ్ హై పెనిట్రేషన్ కెపాసిటీ. అర్థమైందమ్మా ఇప్పుడు గుర్తుపెట్టుకోవడానికి? నౌ these X-rays, these gamma rays, infrared rays and X-rays, they will destroy the cell wall of the they will destroy the cell wall of the bacterial cells as well as their ribosomes, అంటే ప్రోటీన్ సింథసిస్ ఆపుతుంది. కానీ యూవీ రేస్ ఏం చేస్తాయంటే మైక్రో ఆర్గానిజంలో దే విల్ డ్యామేజ్ ది న్యూక్లిక్ యాసిడ్స్, దట్ ఈజ్ ది DNA. ఇన్ ది DNA it consists of adenine, thymine, cytosine, guanine. Adenine always binds with a double bond with thymine and cytosine always binds with a triple bond with guanine. Now what happens is these UV rays, they will start, suppose here thymine is there, here adenine ankondi, these two thymines they will start binding with each other. The adjacent thymines they get bind with each other and these bonds will be broken. When these bonds are broken between thymine and adenine, these two adjacent thymines they form a thymine-thymine dimer. A thymine-thymine dimer is formed so that the bond will be broken, distorted, and these radiations they destroy or kill the microorganisms. Okay, this is about your radiation. Coming to the next one, that is your sixth one, is your hydrostatic pressure. What is hydrostatic pressure? The pressure that is present underneath the oceans, at the deep oceans. If any pressure is there, that pressure will destroy the microorganisms. చూడండమ్మా, if submarines are going, మీరు ఘాజీ అనే సినిమా చూసుంటే కనుక, చూడకపోతే చూడండి, దానిలో ఏమవుతుందంటే సబ్మెరైన్ లోపల ఉంది, ఆ సబ్మెరైన్ లోపల వాటర్ లోపల ఉన్నప్పుడు అది ఎంత లెవెల్ ఉండగలదో అంతే ఉండగలదు. ఇంకా కిందకి వెళ్ళింది అనుకోండి, అక్కడ హైడ్రోస్టాటిక్ ప్రెషర్ పెరగడం వల్ల ఇమీడియట్గా ఈ సబ్మెరైన్ రెక్కేజ్ అయ్యే ఛాన్స్ ఉంది, అంటే బ్రేక్ అయ్యే ఛాన్స్ ఉంటుంది. అంటే ఏంటి అంటే దో సబ్మెరైన్స్ దే విల్ మూవ్ అండర్నీత్ ది వాటర్, దే కెన్ గో ఓన్లీ అప్ టు సర్టెన్ డెప్త్. When they enter, they go to the bottom, try to go to the bottom, immediately due to this high hydrostatic pressure, the breakage of that submarine takes place. That means, see here, its hydrostatic pressure here underneath the ocean, that is at the bottom of the ocean, deep oceans, they have a very high hydrostatic pressure, that is 1000 times more than the hydrostatic pressure that is present on the seen on the surface of the water. అంటే ఏంటి, సర్ఫేస్ ఆఫ్ ది వాటర్లో ఎంత ప్రెషర్ ఉందో దానికన్నా 1000 టైమ్స్ ప్రెషరు కింద ఎక్కువ ఉంటుంది. అందుకోసమని ఈ మైక్రో ఆర్గానిజమ్స్ ఏవైతే కింద ఉన్నాయో దెర్ ఇస్ ఎ ఛాన్స్ దట్ దేర్ బాడీ విల్ బ్రేక్ ఇంటూ పీసెస్. దేర్ బాడీ విల్ బ్రేక్ ఇంటూ పీసెస్ బికాస్ ఆఫ్ దిస్ హైడ్రోస్టాటిక్ ప్రెషర్. అందుకోసమని ది మైక్రో ఆర్గానిజమ్స్ దట్ గ్రో in the deep oceans, they are called barophils. What is barometer? All of you know that barometer is to calculate the hydrostatic pressure. The instrument is called barometer. So these organisms, they are called barophils, that can grow at high hydrostatic pressure areas, or they can grow in the environment consisting of high hydrostatic pressure, that is at the bottom of the oceans. The microorganisms are the organisms that are growing at the bottom of the oceans and sea and oceans. They are at the bottom of the sea and oceans, they are called as barophils. Now coming to the last one, that is your water availability. See here, coming to the next one, that is your water availability. What is water availability? Water availability is also known as water activity. In other words, it is called as water activity. That means water will play an important role in the growth of the microorganisms. All of you know that if there is moisture, the microbial growth will be more. అంటే రిలేటివ్ హ్యూమిడిటీ ఎక్కువ ఉంటే అక్కడ మైక్రోఆర్గానిజమ్స్ గ్రో అవుతాయి. In the last class I told you that during rainy season and winter season, the microbial growth will be more because moisture is there, relative humidity is there in the atmosphere environment. And next, in the summers, due to dryness, the microbial growth will be less. So here also, due to water availability or water activity, the microbial, due to presence of relative humidity, the microorganisms, their growth is affected. What is their growth? Due to the presence of water, the microorganisms will grow. This is first one. Second one, based on the water availability, the microorganisms are of 3 types. They are halophils, all of you know that, osmophils, xerophils. See here, A is osmophils, A is halophils, B is osmophils, C is xerophils. What is this halophil? Halophil already we have studied here. Halophils are the organisms which grow at high salt concentration, and osmophils are the organisms that grow at high sugar concentration. That means they will be taking salt and mixing it with water. In that water consisting of high salt content, the organisms will be growing. We are taking water, and in that water the sugar is mixed, and in that sugar concentration, concentrated solution, in that high sugar concentrated solution itself indicates that it is liquefied, that is it consists of water. So osmophilic, the microorganisms grow, that those microorganisms are called osmophils. Now coming to the next one, that is xerophils. These are the organisms which grow in the absence of water. That is the reason it is called as xerophytes. What are xerophytes? These are the plants that grow in the absence of water in the deserts. Example is your cactus. The cactus plants, they grow in the absence of water in the dry environment. So in the atmosphere, whatever oxygen is whatever water is there, that will be absorbed by the plants and they will grow. Why? Because in deserts during night time it will be highly cold. Due to that, some water content will be there, and that water content will be absorbed by the plants, and these plants, they will grow. They are green in color only because of the availability of oxygen. They undergo photosynthesis and they produce starch by means of photosynthesis. So chlorophyll is there, so they can grow in the desert areas though water is not available in the soil. So These xerophytes are the plants that grow in the absence of water in the deserts, or in the, you can't say that in the absence of water. Xerophytes are the plants that grow in the deserts, in the desert areas where water availability is very less or least amount of water, less amount of water availability is seen. Xerophils are the organisms which grow in the dry areas. Such type of, that means here the water availability is not there, or water activity is not seen. So xerophils are grown. That means whatever it may be, water is an essential factor for the growth of the microorganisms, or not only microorganisms, for every, or for any organism, water availability or water activity is must. Why? Because all our cells, we have 90% of water in the cytoplasm of the cells. That means all our cells, in all our cells, the cytoplasm consists of 90% of water. So these are the factors that are responsible or that are affecting the growth of the microorganism. Okay students, we will stop here, and in the tomorrow's class we will continue with the next topic. Thank you students.